There are actually several different types of mutations that we find in microbial cells, and in fact in all living cells. We can categorize those mutations as being either point mutations or what we call chromosome level mutations. Point mutations, like the name suggests, are mutations that occur at just a point in the DNA. In other words, a single base is in the wrong place, is out of order. Chromosome level mutations are very different. Chromosome level mutations are the kinds of mutations that are caused when entire sections of chromosomes get broken off or get broken off and then reinserted in a new arrangement. Chromosome level mutations are actually quite common in human cells and in other higher eukaryotes, but it's point mutations that we see primarily in microbial cells, and so that's what we will focus on in this lecture. So there are 4 specific point mutations I want us to be familiar with. The first kind is called substitution. So when you hear the word substitution mutation, I want you to think that substitution means switching one base for another. So maybe in the old DNA there was a G in a particular location, and in the new DNA, instead of putting a C there, the enzyme puts either an A or maybe a T. That's a substitution mutation. One base has been switched for another. Now, a substitution mutation can change the protein that's made from that DNA. It's just that it doesn't always change the protein. Remember, for each of our amino acids, for each of the 20 amino acids, there are multiple codons that code for it. Sometimes there are as many as 4 codons. And so it's possible for a mutation to occur, changing a codon, because all mutations change codons, but the codon isn't changed quite enough to change which amino acid gets placed there. So we have to remember that substitution mutations sometimes change the order of the amino acids and sometimes they don't. Silent mutations are actually a type of substitution mutation. We still see one base being switched for another, but in a silent mutation, the swapped base does not lead to a different amino acid being placed in that position. So this is a case in a silent mutation where the codon changes due to the mutation, but it doesn't change enough. It changes to just a different codon that codes for the exact same amino acid. So there's no change in the protein and no change in the trait. It's silent, in other words. We would never know that the mutation was there because there's no change in the protein and there's no change in the trait. The next point mutation is called a nonsense mutation. And in this case, a substitution mutation takes place. One base is swapped for another, but the codon now becomes a stop codon. Remember that a stop codon is a codon that tells the ribosome to stop translating at that spot. The start codon is the codon that tells the ribosome to begin translation, and the stop codon tells the ribosome to stop translation. So if a mutation occurs in a codon that would normally code for an amino acid, and the mutation causes that codon to now be a stop codon, we've got a huge problem because that stop codon is premature. It's in a place where it shouldn't be. It's going to stop translation, and we're going to end up with a protein that is much shorter than it should be. It doesn't have the right number of amino acids in it. So that's going to change the trait severely, and sometimes to the detriment of the organism. Finally, there are what we call frame shift mutations, and these are not substitution mutations, because instead of swapping one base for another, in a frame shift mutation, either an extra base is added in or a base is removed entirely. That's a very important change, a very important difference from a substitution mutation. We're either adding in an extra base or we're removing a base entirely. That causes the shift in the reading frame. It causes a shift in all of the codons from that point forward. Imagine if you have codons, groups of 3 bases, one after the other. If you add in an extra base, you've now shifted every codon from that point on by 1 base. If you remove a base, you've now shifted every codon from that point on by 1 base. So just like nonsense mutations can cause big changes in the protein, frame shift mutations do the same. They change a whole bunch of codons, which changes a whole bunch of amino acids and causes a massive change in the protein. Now, I want to change gears a little bit here and talk about a really important laboratory technique called polymerase chain reaction, or PCR. PCR has been in the news because the primary test that is used to determine whether or not a person has the new coronavirus is a PCR test. It looks for the genetic material that's inside this virus, and it can detect whether or not a person has the virus by detecting its viral genetic material. Polymerase chain reaction is a very common technique in medicine, and it's becoming more common every day. So we need to have an understanding of how it works. And the reason we're talking about it today is because it essentially is a copy of what the cell does during DNA replication. In essence, it's DNA replication done in the laboratory, or done in vitro, as we say. This phrase, in vitro, translates to in glass. And the way we interpret it is that we are doing something outside of the living cell. So when something's done in vitro, it's done in the laboratory instead of inside a living organism. So polymerase chain reaction is just DNA replication done in vitro. A region of DNA that we're interested in, usually it's a gene, gets replicated over and over and over again during PCR. And the term we use is amplified. We're going to amplify a piece of DNA. And the reason you would want to amplify a gene or amplify a piece of DNA is because once you have many copies of that piece of DNA, you can do things with it in the laboratory. And one of the most common things that's done with PCR is we take that amplified gene and we sequence it. We determine the order of the bases in that gene. In the case of the coronavirus, we amplify the genetic material that's present in the virus, and we look for it on what's called a gel. we take all of those amplified pieces and we put them inside an agarose gel, and we can visualize the DNA inside this gel. So PCR helps us diagnose things in medicine, and it helps us learn about genes in the laboratory. PCR literally creates millions of copies of the piece of DNA that we're interested in. And it's in order to create those millions of copies, it performs replication over and over and over again. It's a very clever process, a very clever reaction. Now, in order to do DNA replication in PCR form, you have to have a very special type of DNA polymerase. And the type that's used for PCR is called Taq polymerase, TAQ. There is in 2020 a machine that does PCR for us, and we call that machine a thermocycler. So, polymerase chain reaction, or PCR, takes advantage of the complementary nature of DNA, just like the cell takes advantage of that to do DNA replication. Remember, inside the living cell, the old strand of DNA is used as a template, and the DNA polymerase enzyme builds new DNA on top of that template. The same thing is going to happen during PCR. We're going to take a double-stranded molecule of DNA and the Taq polymerase enzyme, and we're going to copy that DNA over and over and over and over again until we have millions of copies of it. Now, the way DNA replication happens in a living cell, as we've discussed, involves lots and lots of different enzymes. The way it happens in PCR involves temperature changes. It's a very clever system. The first step involves denaturing the double-stranded molecule of DNA. We heat up the reaction tube to a very high temperature, about 94 degrees centigrade, and that causes the double-stranded DNA molecule to separate into two strands, or denature. In step 2, we reduce the temperature inside the test tube, and we allow the primers to adhere to each strand of DNA. Remember, in a living cell, when the DNA strands are separated from each other, an RNA primer gets built on each of the strands. During PCR, we're not going to build the primer in the reaction. We simply add preformed primers into the reaction. This process is where the primers attach to the old DNA is called annealing. The individual primer for each of the old strands of DNA will anneal with that strand or attach to that strand at a temperature of about 54 degrees centigrade. So that's still a pretty hot reaction at that point, but it's a lot less hot than what we started out at. Step 3 in the PCR is called extension. We raise the temperature to about 72 degrees centigrade, and that happens to be the temperature that Taq polymerase functions optimally at. So 72 centigrade is the optimal temperature for Taq polymerase to build new DNA. We allow that extension phase to go on for a little while, and then we repeat the process. We heat the reaction up again, and we denature. We cool it off a little, and we anneal. Then we raise the temperature up a bit, and we extend. And then we repeat it again. And it gets repeated over and over and over again, usually between 30 and 40 times. Each one of those times is called a cycle of PCR. So most PCR reactions include 30 to 40 cycles. Now, we've done what a cell does with enzymes. We've done with temperature in PCR. And that's very clever, but it causes a problem in that an ordinary DNA polymerase enzyme would not be able to survive a reaction tube that was heated up to 94 degrees centigrade. That's why we need this special kind of DNA polymerase in PCR. You see, Taq polymerase comes from an organism, a microbe, that lives at the bottom of the ocean inside thermal vents. So there are these vents on the floor of the ocean that have hot gases coming out of them from the center of the earth, and those vents reach enormously high temperatures. So there is an organism called Thermos Aquaticus that likes to live inside these thermal vents. Now you can probably guess that Thermos Aquaticus is an archaea. It's an archaeal organism. It's an organism that lives in an extreme environment, extreme heat. So we have isolated Taq polymerase from Thermos Aquaticus. That's where the name comes from. The T from Thermos and the AQ from Aquaticus. That's Taq polymerase. And the reason we need the Taq polymerase is because we're going to heat our reaction tube up to 94 C. And this enzyme has been adapted to those kinds of high temperatures. So it'll work for us inside our PCR reaction. Very clever. This last slide demonstrates for us how 30 to 40 cycles of PCR can lead to millions of copies of a piece of DNA. Up at the top of the slide, you can see a drawing of the 3 stages of one cycle of PCR. Remember, we've got a piece of DNA that has a gene in it that we're interested in. So it's a double-stranded piece of DNA. When we put it into the reaction tube, we're going to heat it up to 94 degrees centigrade, and that's going to cause those 2 strands to separate. That's denaturing. Then we cool the reaction down to 54 degrees, and that allows the primers to anneal to each strand. Remember, we're putting the primers into the test tube. Then we heat the reaction tube back up to about 72 degrees centigrade, and we allow extension to take place. The Taq polymerase enzyme is going to start adding bases one at a time into the new DNA strand using the old DNA as a template. So by the end of one cycle, we've taken one double-stranded piece of DNA and we've turned it into 2. Now look at the bottom part of the diagram. This is the part that shows us the amplification part of PCR. Again, here's our original piece of DNA shown in red. We go through one cycle and now we have 2 pieces of DNA. We go through a second cycle and each one of these is going to get replicated. So this one gets copied into 2, this one gets copied into 2. Now we have 4. We go through another cycle and now we have 8. And then another cycle and now we have 16. And then 32, and then 64, and on and on and on. This is an exponential reaction. It creates an amplification of that original piece of DNA. By the end of 30 to 40 cycles, you literally have millions of copies of your original piece of DNA.